Hi everyone, it's Dr. LeBlanc with Unit 13A, Linkage Mapping and Aneuploidy, Linkage and Recombination. Our learning goals are to learn that the recombination frequency between genes on the same chromosome is related to the physical distance between them, to learn how to calculate recombination frequency by counting phenotypes of the outcomes of crosses, and to learn how to determine the order of genes on the chromosome. Let's start with a revisiting of Mendel's independent assortment law, which in a dihybrid cross yields a 9:3 to 3:1 phenotypic ratio. Let's start with some pea plants. Some tall, yellow seeded pea plants crossed to some short green ones. In the P1 generation, the parents are true breeding. They are homozygous for the dominant alleles in this parent and the recessive alleles in this parent. And when their offspring are produced, they all appear tall and yellow, but they are actually heterozygous at both of these genetic loci. If we cross our F1s together and generate an F2 generation, we can determine what the phenotypes are by filling out a Punnett cross. Remember, in a Punnett cross, across the top are the genotypes of the sperm, and on the side are the genotypes of the eggs. Sperm and eggs are haploid, so they have only one allele for each of the two genetic loci that we're following. So we have a tall allele here and a yellow allele here in this sperm, and the same for this egg. And when we mix those two together, we end up with a diploid individual with two alleles for the tall gene and two alleles for the pea color. If we look at the ratios of the offspring, if we look at the genotypes and the phenotypes, we find that we have 9 plants with the two dominant phenotypes, tall and yellow, and you can have various combinations of alleles to produce that. We have 3 plants with green seeds who are tall. We have 2 different genotypes that produce that. 3 dwarf, which is the recessive phenotype, with yellow seeds, the dominant phenotype, and then 1 plant that has both recessive phenotypes displayed, this one right here. Okay, that's what happens if the genes are assorting independently, if they're not linked together. Let's see what happens with these flowers. We take a purple flower with long pollen grains and cross it to a red flowers with round pollen grains, little ones. The F1 offspring are all purple with long pollen, so we determine that those are the dominant phenotypes. If we allow the F1 offspring to self-fertilize and those two genes are assorting independently, we would expect a 9:3 to 3:1 ratio. However, that's not what we get. Instead, we get something very different. We have a couple of phenotypes that are quite prevalent and 2 other phenotypic combinations that are not as prevalent as the others. This one and this one are much greater proportion of the 2 types found in the parental generation, purple flowers with long pollen and red flowers with round pollen. That is a definite giveaway that those genes are linked together on the same chromosome. Remember that during meiosis, you start out with a diploid cell, and then the sister chromatids separate to form haploid gametes. If there's no genetic recombination between these, no crossing over, each cell of each one of these 4 cells will end up with the genetic combination that was found in a parent, either the 2 dominant alleles from this chromosome or the 2 recessive alleles from this chromosome. The haploid cells contain the same combination of alleles as the original chromosomes, and because they're linked and they haven't been altered. Now, what if we do get crossing over in between the A and the B genes? After crossing over, then we can end up with different combinations of alleles than what was present in the parent. We still have chromosomes that have the same combination, the 2 dominant alleles on one chromosome and the 2 recessive alleles on a chromosome, but we also get a recombination of that. We have one dominant and one recessive and one dominant allele now. These two cells are the recombinant cells. They contain a combination of alleles not found in the original chromosome. The new combination is the result of genetic recombination, and these are termed the non-parental or recombinant, whereas the other two are called parental or non-recombinant. Okay, what about the fly? Thomas Hunt Morgan provided a lot of evidence for the linkage of several X-linked genes. He's followed many different phenotypes that were mapped to the X chromosome. Here we have a female fly that is yellow-bodied, white-eyed, and has miniature wings. And if we cross her to a wild-type male that has a gray body, red eye, and normal wings, we end up with not one single dominant phenotype, but we end up with a reversal of the sexes. So that now we have, in the F1 generation, males that resembled their mothers and females that resembled their fathers. And if we count up the flies, scoring them on the presence of those three different phenotypes, we see that there are mostly gray-bodied, red-eyed, long-winged flies in the F2 generation, and also mostly yellow-bodied, white-eyed, miniature-winged flies in both male and female. Those are the two most abundant phenotypes, phenotypic combinations. And then we also have other recombinant mixes of those phenotypes, a yellow body and white eyes, which are recessive, plus long wings, which is a dominant trait. And we also have gray body and red eyes, which are the two dominant phenotypes, plus a miniature wing, which is a recessive phenotype. Notice that these two are the reciprocal of each other. And then we have other combinations that are far less common. Although the flies with the parental traits were the most common, there were flies that were obtained that had recombined parental traits. Some recombinants were more common than others. There was actually some cytogenetic evidence that crossing over results in recombination of alleles. Harriet Creighton and Barbara McClintock worked with corn, and they found this very abnormal looking chromosome that they could actually visually see under the microscope. It had a little knobby here and a little piece of chromosome 8 translocated. This is what a normal chromosome 9 looks like. When you get crossing over, you get some even more visually detectable combinations of chromosomes. These are the non-parental ones. These were the parental ones. Exchange involving the normal and abnormal chromosome 9 yields chromosomes that were different from the parents and that you could visually see under the microscope. Then this was due to crossing over, breakage, and recombination. So when McClintock followed the genes that were located on these chromosomes and could visually see them under the microscope, they were able to determine that the physical exchange of these genes also resulted in recombination of different phenotypes. How can we use recombination in mapping? Genetic mapping in plants and animals involves first establishing linkage groups. Linkage groups are a functional equivalent of a chromosome. So all of these genes are functionally linked together and physically linked together on the chromosome. One important cross that we need to perform when we're using recombination to map genes is a testcross. In Drosophila, males do not undergo genetic recombination during meiosis. It just doesn't happen. The only meiotic recombination that occurs is in female flies. So here's an example of a test cross where we have a heterozygous female parent for short bristles and ebony body. She is heterozygous. She carries both alleles, but the dominant phenotype are long bristles and gray body. We test cross her to a male that is homozygous recessive for both of those traits. So this male has short bristles and an ebony body. Remember, the plus indicates wild type. When we count the progeny from this cross, we have some recombinants. These chromosomes are the product of crossing over during meiosis in the heterozygous parent. So if we get crossing over here, we can mix a wild-type bristle allele with an ebony body allele. That would be this, wild-type bristle and an ebony body. This fly is going to have an ebony body and normal bristles, and this is the reciprocal. This is going to have a wild-type body and short bristles. These are the unrecombined parental combinations of those alleles. So if they were not linked together, we would see a 1-to-1-to-1-to-1 combination ratio of all of these different phenotypes. But because they are linked together, we see more of the non-recombinant parental types and fewer of the recombinant ones, which are the product of crossing over. Now, here's how we can use the recombinants to map genes, the distances between genes. The number of recombinants are used to calculate a relative distance between the genes. The map distance is equal to the number of recombinant offspring divided by the total number of offspring times 100. So we have 76 plus 75 divided by all of the flies, the total number of flies, multiplied by 100, and that gives us 12.3 map units or centimorgans. These are equivalent units. That's not too hard. Okay, now we need to be able to order genes along the chromosome and determine the distances between each of those genes, so that we can start to build up a genetic map by continuously adding different genes to a trihybrid cross. It's a very common strategy. This example uses 3 genes on the same chromosome for body color, eye color, and wing shape. For body color, the mutant is black and the wild type is gray. For eye color, the mutant is purple and the wild type is red. And for wing shape, vestigial is the mutant and normal wings or long wings is the wild type version. So what do we start with? We start out with a female that is black-bodied, purple-eyed, and vestigial wings. And we made her to a wild-type fly, male. When we make these two together, we end up in the F1 generation. Female flies that are wild type in appearance, but we know that they are heterozygous for all 3 of those genetic loci. Wild type recessive, wild type recessive, wild type recessive. Then we take her and mate her to the test cross male, which is the double homozygous recessive for each of these 3 genetic loci. We need to perform the test cross by mating the F1 female heterozygotes to the males that are homozygous recessive for all 3 alleles. Now remember, crossing over can occur in the female, but not in the male. Even if it could occur in the male, it wouldn't make any difference as far as these 3 genetic loci are concerned because they're homozygous at all 3 of these loci. So we wouldn't be able to detect recombination using just those loci. Okay, Then you mate those flies together and you collect the data for the F2 generation for a total of 1,005 flies. If the genes are sorted independently, all 8 combinations would occur in equal proportions. But in the offspring of the crosses involving linked genes, the parental phenotypes occur most frequently. Okay, so here's our list of flies with the varying phenotypes. Here is our most common, and right here, these are the unrecombined parental phenotypes. So we have the mutant black body, purple eye, and vestigial wings, and then we have another chromosome with all 3 of the dominant alleles here. So these are the unrecombined parental chromosomes. Double crossover phenotypes occur the least frequently. So which are the double crossovers? It's this one and this one. So if we look at the double crossover, we can determine which gene is in the middle. The wild type for body color and wild type for vestigial were together in the initial parental chromosome, but here they're combined with a purple allele. So purple is going to be in between the body color and the wing shape. Let's look at if the reciprocal is also true. Let's look down here. Okay. Here we have the recessive allele and the recessive allele, but the wild type allele for purple. So the odd man out here, the one that's different in the combination between the other two, is the allele, is the genetic locus in the middle. And single crossover phenotypes occur with an intermediate frequency. So here, 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 and here are all the single crossover events. So here we have the black allele, the wild type allele, and the wild type purple with the recessive vestigial. And down here we have a different recombination event. We have the wild type for body color, but the purple eye and the vestigial wing are the mutant phenotypes. Okay, so let's put some of these numbers to work. We're going to calculate the distance between pairs of genes. We separate the data according to pairs of genes and add the number of flies for each pair that do not have the parental combination, i.e. the recombinants. We're going to add the number of recombinant flies together. For example, for body color and eye color, the recombinants are the gray body is the wild type, plus purple eye was the mutant, so that was a recombination. There were 30 flies and then 2 other flies. And then for black body with red eyes, we had 28 plus 1. So we add all those numbers up together. We divide by the total number of flies, multiply by 100, and we get 6.069 map units or 6.1 map units that are between the B and the PR genes. This is the map distance between the body color and eye color genes. The map distance between eye color and wing shape is, let's see, eye color is the purple and wing shape is vestigial. So here's a recombinant between those two. This was wild type, and this was vestigial. This is the recessive, which is different from the parental combination up here. So here's a recombinant. Here's another recombinant. This is not a recombinant. These are the same as a parental chromosome. This is also the same as a parental chromosome. This is different combination than a parental chromosome. And this is also different. So it's 61 plus 60 plus 1 plus 2 divided by 1,005 times 100 equals 12.3 map units. So that's the distance between the purple and the vestigial genes. The map distance between body color and wing shape spans this whole genetic region here that we've been discussing. We have to remember that when we count these, the double recombinants, we have to multiply by 2 because there were actually 2 recombination events to produce this combination of alleles. So even though the recessive allele for body color and the recessive allele for wing shape was the same allelic combination as what we saw in the parental chromosome, because it has the wild type allele in between these 2 recessive, we know that it has been 2 genetic recombination events. So we have to count these double. So we have to add those to all the recombinants. So we have 60, this is 60 plus 61 plus 28 plus 30 plus 2 times 2 plus 1, that's this and this, the double recombinants, yields 185 divided by 1005, or times 100 gives you 18.4 map units. So here's our map. We have 18.4 map units between vestigial and black, 6.1 map units between black and purple, and 12.3 map units between purple and vestigial. And you can double check to see if you did everything right, because this distance plus this distance should equal this distance. The data is also consistent with the map being drawn opposite, with the vestigial here and the black here. In detailed genetic maps, the location of genes are mapped relative to the centromere.